Thank you. So thank you, Peter, for that kind introduction, and thank you for the gracious invitation to come here. And I've had a wonderful time so far, and I'm looking forward to the next um, almost 24 hours. Hopefully, the snow holds off, but it doesn't look very promising. But I'm sure you can deal with it. So probably better than people from Toronto. So, yeah, as Peter said, I come from the Ontario Institute for Cancer Research, and what I wanted to do, I debated whether to go into a more in-depth treatment of a particular project that we worked on, but I said instead to kind of give you a smorgasbord of different projects, hoping that there would be something of interest for everybody. And before I even go into the projects and the science themselves, the science of the projects themselves, I wanted to give you a little overview of our group. It sounds a little bit like an advertisement, but one of the key things I'm excited about in coming here is the opportunity to be able to collaborate with people here. And, if I can show you a little bit about who we are and what our capabilities are, perhaps that would stimulate some thoughts into how we could possibly work together in different projects. So after I tell you about that, I'll go into the three projects that I want to tell you about. They're all kinase projects, and so far I've heard a lot about kinases already today. So you'll have to pardon the very basic introduction because I wasn't sure what the level of expertise across the audience was. But I'll introduce kinases, and then I'll tell you about the three projects that we're working on and that I want to tell you about, and then finish up. And so just an overview of who we are and what we do. This is a series of chevrons, which you probably already all seen in some way, shape, or form. Basically, that talks about the whole drug discovery continuum from start where a biologist will identify a potential target of interest that's related to, in this case, oncology, and then various scientists with different skills and disciplines come into the fore to develop and prosecute it to find a lead compound that is then taken into clinical development. And where we tend to play is at the very early stages of the whole process where we have a set of biologists, and I'll tell you a little bit more about who they are shortly, but they are very involved with target validation and identification. And so oftentimes when we collaborate with academic scientists, they have a great idea, but maybe it needs a little bit more validation in the context of a particular tumor type. And we have a group of biologists that will help there. And then, of course, we have medicinal chemists, and that's how our group started to help with once a screen is set up and is shown to be operating on high throughput level or medium throughput, as the case may be. Then we can supply compounds for screening, and we can help triage the hits for further validation and identification so that we can go into the more advanced stages of the project, which is hit to lead. And then once we get a valid lead compound, our typical funding envelope stops, but that's where, and you see the kind of the red checkered box, that's where we really seek partners that have deeper pockets, so to speak. So people from pharmaceutical companies, people with bigger amounts of money to spend that helps do the more expensive lead optimization phase of the project. And then beyond that, we're looking for partners to take our lead compounds into development. And so we've had a couple of different examples where we've worked in major pharmaceutical companies to do that lead optimization stage, but typically where we play is in the blue box. And so I'm a part of a group, and it's a little bit complicated to explain, but drug discovery is a subset of a broader group known as Therapeutic Innovation at Ontario Institute for Cancer Research. And our mission is to take novel validated cancer drug targets from academic researchers and from our own group, and to help discover first-in-class or best-in-class drug candidates that can be taken into preclinical development. We want to ensure those drug assets have a competitive target profile, which is easier when you're first-in-class than it is when you're best-in-class. But nonetheless, we want high-quality molecules that serve as drug candidates and then can be invested further into late preclinical development and then clinical testing ultimately. And so we have three different programs within therapeutic innovation. And one of them is drug discovery. And that's really the infrastructure of the group that I'm going to tell you about, where we do all the different assays and the lead medicinal chemistry and all the analytical chemistry. And then we have an aspect of it, which I've been telling some people about, and I'd like to talk to more people about here, is our collaborative research resources, which is basically how we can help very early on when projects are at the early stages, where we could do not necessarily extensive research, but key experiments that we can help further the project along so that it can actually be a good funding opportunity. And of course, we would encourage our collaborators to apply to what's called the CTIP, or the Cancer Therapeutic Innovation Pipeline application. And this is a funding envelope that I'll tell you a little bit more about. And we also have the drug discovery community, which is another thing that I've mentioned already to some of you today. And that is basically we are going out. It's an outreach program where we're looking for promising research, promising investigators in which we can continue to help them in terms of moving a project along in their own research and possibly into our pipeline. And so the Cancer Therapeutic Innovation Pipeline that I told you about, this is a very advanced funding mechanism, and this is a little bit complicated, and I don't want to go through, in the interest of time, all the aspects to this, but basically we have different phases of funding where a researcher can apply at a very beginning and have their target be further validated, and then it can go into what's called the early accelerator program, where you're actually trying to develop a screen for for high throughput screens or for whatever type of screen is needed, and then you go into a late accelerator program where you're actually looking at trying to identify a validated hit compound that can be further optimized. And so we have all kinds of projects which span the continuum of these different phases. And so, as I said, a researcher can apply to any of these phases, but we have what's called a TPAC, which is a committee that sort of oversees all of the research in this funding envelope. And not only do they give advice in terms of monitoring the progress of projects, but they also can give guidance in terms of what phase is worthwhile to apply to. So this is something that's on our website, and I encourage you to look at that if you have a project which is of interest for moving forward and you would like to potentially work with us. I will say that this funding is actually independent of my group, and you can actually you can get funding and you can do all of this research outside of our group. Of course, I would love it if you would collaborate with our group in this work. So the drug discovery community, just to say a little bit more about that, it's really spearheaded by Jay, who's in the middle, the picture in the middle, and the whole idea is to develop a unified collaborative community that networks drug discovery groups across the province, such as here at Queen's, to strengthen the capacity and leverage resources to develop homegrown therapeutics, so that you wouldn't necessarily have to collaborate with somebody in the U.S. or somewhere else, but it's really a whole idea is to strengthen the provincial research. And what we have is are these drug discovery days where we actually can go and have a focused set of presentations from different groups at that institution. And if there's a critical mass, we will actually organize a drug discovery day. And so that's something that I would like to explore further with some of you, is whether we can actually have, if there's people here who have projects that you think are relevant for the Cancer Therapeutic Innovation Pipeline or to work with us in general, we would love to actually organize an event where we come out and we talk to people. And that meeting that I'm describing will actually involve our therapeutic pipeline committee, our TPAC committee, and they will actually give advice and feedback into the projects to help move them along. So that's something that we can continue to explore. Here's our leadership team, and I just briefly highlight the different people. Starting from the lower right is Dr. Ahmed Aman. He's a person who does our DMPK and analytical chemistry. So, for example, if you're interested in understanding whether your drug is getting on board prior to doing an in vivo efficacy study, he would be a person who would be able to help out using mass spectrometry and other advanced instrumentation techniques. I talked about Jay Ramnath. He's kind of the glue that holds our group together in many ways. He has a lot of industry experience, and he also is very good about helping manage collaborations, and he's often the first point of contact in our group. Um, uh, Going back to the bottom, Dr. Methvin Isaac, he's a very seasoned medicinal chemist, and oftentimes he'll be finding himself in a leadership role in different projects that we work with. And going up again, I'm going zigzagging through this group, but Dr. Masood Badadi, he is actually the latest addition to the team, and he comes to us from the Structured Genomics Consortium, and he's an expert in protein production and X-ray crystallography, and has actually had some breaking publications. One of which recently appeared in Nature, describing the ternary structure of a PROTAC from a new E3 ligase known as DCAF, which I won't be telling you about today, but I think he's been a tremendous resource for our group. Dr. Gena Poda, back down on the bottom, is our lead computational chemist. He also is well versed in AI, and is helping lead a team that is actually looking at spinning off a company called QXL for artificial intelligence, and is also very interested and very collaborative person who has helped a lot of different PIs throughout the province in terms of applying computational chemistry and molecular docking and high throughput virtual screening. And then back at the top is Dr. Richard Marcellus, who's the senior advisor of biology. So he's our key biologist who is leading our cellular biology groups, but also he's a real expert in SPR. So he's been leading a lot of he and his group, I should say, have been leading a lot of our high throughput screening efforts. You've already heard about me, but then last but not least is Dr. Rima Alawar. Rima was the senior leader of the group, and she's recently left for Oman, and I'm taking over her role, but we're fortunate that we still have her as an advisor. So she's been great. And I was joking to some of you that it seems like she left and went to Oman, and I've actually had more talks with her since she's left than during the time she was actually at OICR, but it's just because she's been very active and involved with some of the projects that are near and dear to her heart. And of course, the group is near and dear to her heart. So this is our a slide that just basically highlights the capabilities of our group. And we have medicinal chemistry, cell biology, computational chemistry, protein structural biology, ADME, and analytical chemistry, and of compound screen development. We have compound libraries that I'll tell you a little bit about, and formulation and efficacy studies. And so we have a lot of individuals who are from academics, straight out of academics, but we also have a lot of people who are from industry. And so we've had kind of a strong industrial background, which I think in sort of the academic setting makes our group, if not unique, at least distinct, especially here in the province. And so our role is really to bridge the gap between academia and industry. And so I just wanted to tell you a little bit about some of our scientific areas of interest. And this is by no means all encompassing, but it's some of our key emphasis. We look at, we have kinase projects, and so we have the so-called kinase platform. And I'll tell you more about that later in terms of our science. We have a WDR platform. WDR proteins are a very interesting novel class of proteins, which we've really had a long-standing interest in. And of course, anything that's related to WDR, we've worked very closely with the Structured Genomics Consortium. And then we have a strong interest in immuno-oncology, which is why I've been delighted to hear some of the targets that Peter's been involved with. But supporting all that is some different areas of interest in terms of protein production and structural biology, computational chemistry. We have a new emerging platform in lipidomics. which is something I can tell you about more offline. And then PROTACs, we're very interested in PROTACs, and we have novel linkers and ways of hooking on our activating molecules to the E3 ligases. And then our newest area that we're really heavily involved with is radiodiagnostics and theranostics. And we have a very active, we're fortunate to have such a strong expertise. You probably heard of Fusion Therapeutics, which recently went public. That OICR actually helped fund Fusion at a very early stage, and through the Center for Probe Development and Commercialization, CPDC, running out of McMaster University, we actually have a strong collaboration with them. And so any project that we work with, any collaborator that has the potential to be developed into a radiotheranostic or a diagnostic, we can work with them on, and they have the expertise. They have the nuclear chemistry expertise to help make that realization. And then, of course, supporting all of that is medicinal chemistry, our cell biology and biophysical chemistry, and our ADME and PK. So in terms of the compounds that we have available, we have compounds that are sitting within our group, collections that is, but we also we've been able to try to leverage these compounds by housing them in different screening centers. And all of these are within the local Toronto area, but they have, of course, anybody in the province has access to these screening centers. And so without going through all of these in detail, we have a high throughput screening compound collections of over 100,000 compounds. We have a very interesting novel set of molecules from chem routes, which is a little over 20,000 compounds. We have a 24K diversity set, which some of you have already screened against, and then various other different collections, including fragment collections and other collections, natural product-like collections as well. And one collection that's most relevant to the subject matter that I'm going to tell you about today is our kinase inhibitor collection. That's now 880 compounds. And what we do is we're very opportunistic. If we find compounds that are available in the patent literature or even commercially, we go ahead and try to procure those. And so these use of that library has actually led to the three projects I'm going to tell you about now. The HPK1, which is an immuno-oncology kinase target, and we have a kinase target relative to the Hippo pathway, and finally GCN2. So I'll tell you, after I give you a little bit of background on this kinase library, I'll tell you some brief vignettes on these different projects, and of course, happy to go into more details in the Q&A or one-on-one discussions. So this is a slide which is embarrassingly basic for most of you, but it's geared towards an audience that doesn't know necessarily what a kinase is, which doesn't apply to a lot of people here. But a kinase is basically a protein that is involved with signal transduction by affecting the phosphorylation of a protein of interest. And kinases typically have a relatively conserved overall shape, which consists of an end lobe, a C lobe, and a hinge binding area, which is where the substrate, one of the substrates, which is ATP, sits. And of course, the protein that is phosphorylated also sits near the ATP in the active site. And so many of the kinases many of the drugs that have been discovered, in fact, the vast majority are targeting this active site of a kinase. And we've now recognize that there's over 500 kinases. Many of you have seen this overall kinase tree that shows the different families of kinases and where they sit relative to their homology and perhaps their function. And of course, within the context of cancer, kinases are extremely important because they're involved with signaling transduction cascades that are often dysregulated in cancers and have other implications related to immuno-oncology, which I will cover more today. And it's interesting, I did this search just last year. I had done it several years ago, but I was curious as to how many drugs were kinase inhibitors that were top selling drugs, and this is the diagram right here, which shows the top selling one is ibrutinib, which was as of 2022 over 8 billion. And all of these compounds are billion-dollar drugs, leading to some pretty impressive efficacy, which has caused them to be really have meaningful impact on the patient. And so, of course, we think that there's many more discoveries to be made that will lead to more of these types of drugs. And so, to help us get started in that endeavor, at OICR, we began back in 2009 when I first joined the group to have a systematic effort to acquire a comprehensive collection of kinase inhibitors. And I think a lot of groups, even commercially now, offer kinase inhibitor collections. They're all distinct and they're different, but I think ours has a lot of advantages in terms of what kinds of kinases. And actually, this should be 880, not 769 diverse kinases. But we've managed to make this set of kinase inhibitors available to academic partners who will use them for cell-based or phenotypic screening, and also to identify novel targets and to probe biological hypotheses. And we also have used them as starting points for internal projects, so that we can actually help probe biological hypotheses, much as like our collaborators would, but also for identifying hits for novel targets that we might do for either internal projects or or with our collaborators if they have an interesting target that they want to prosecute. And another useful aspect to having this library is to be able to rapidly. That's one of the beauties of small molecules, and it's very easy to explore combinations and to be able to test hypotheses that involve particular combinations of drug targets. And so this is just an overview of what our kinase inhibitor library looks like. And I don't expect you to be able to see or read the letters, the names on the bottom, but these are basically the names of the different kinases, individual kinases, and the y-axis, the height of the bar, represents how many compounds in our collection are known to cover that particular kinase. And of course, many of these drugs are not selective. Some of them are more selective than others, but the beauty of having multiple drugs from different chemical scaffolds that inhibit a particular target is that it can more easily validate a biological hypothesis. Say if RET kinase, for example, were implicated in a particular cellular phenotype, having multiple RET kinase inhibitors from different chemical scaffolds would be very useful to be able to address that question and see whether that target is indeed relevant to the phenotype. And so, on the pie charts on the upper left basically represent how many compounds are from different kinase families. And on the right, we have, and this is probably a little bit out of date right now, but we try to cover as many clinical compounds as possible and marketed compounds, but also there's a lot of preclinical compounds. And I think that's one of the strengths of this library is we'll go into the patent literature, we'll get compounds that aren't necessarily sold commercially that one can use to evaluate a target. And so using that library, I'll tell you a little bit about three different projects that we've managed to move forward. And the first one is inhibiting a target called GCN2, and that was done in collaboration with Rob Rodapel at UHN. And the angle that Rob brought this project to us through was actually through ovarian cancer, which of course is a very devastating disease. It's the number one killer of all the gynecological cancers, and it's unfortunately mostly diagnosed at late stages, which is one of the reasons why it's so deadly. And its standard treatment is cisplatin, It doesn't seem to be very responsive towards immuno-oncology drugs, at least so far. Standard cytotoxic drugs are the major treatment, and it turns out that cisplatin being the primary treatment is often leads to relapse, and that's a major therapeutic barrier. So high-grade serous ovarian cancer is unresponsive to ICI, as I mentioned, which makes the development of new treatments even more urgent. And interestingly, it's a disease which starts in the fallopian tube and then through the formation of these spheroids that go to the ovary and seed the ovary, and then ultimately form distant metastases. That's what leads to the ultimate deadliness of this disease. And ovarian cancer is associated with the acquisition of new fitness properties and new stress states. And so because there's so many different, it's such a heterogeneous disease, targeted therapy is where you're actually targeting actionable mutations is not seen as a really, has not been at least a promising way to go forward. And so, what we're actually looking at is targeting stress mitigation pathways, because as the disease becomes more evolved, it leads it needs to adapt to these stress states, including genotoxic stress, replicative stress, metabolic stress, and oxidative stress, as well as stress from cytotoxic agents when they're applied as therapies. And so, because GCN2 and the ATF4 pathway is helping mitigate these stress states, we think that that's a promising opportunity to attack ovarian cancer and one of its key vulnerabilities. And so that's the hypothesis that we've been operating with for using this approach. And so GCN2 itself it's a kinase, getting back to the original theme of my talk, but it's also a very important role in sensing amino acid deprivation. And so what it essentially does is it senses uncharged. And when I first heard the term uncharged, I thought it had something to do with a chemical charge, but actually refers to the fact that transfer RNA can be charged with amino acids. And when it's in an amino acid rich state, you have the charging of the tRNA. And when you're in the deprived state, where there's not enough amino acids, you have the uncharged transfer RNA. And that's what GCN2 really senses. And then it becomes activated to signal to the cell, slow down. You need to take it easy, because this isn't a very good environment. So it's really helping the cell survive under these conditions. And this is a slide from Rob Rodapel, who's from California, and he's showing two scenes from California that try to illustrate this principle. But here you have a very lush redwood forest, which is essentially what the cancer cell strives to become. Whereas in a nutrient-poor state, you have a bristlecone pine, which is in a very deprived environment. So GCN2 is essentially shifting the cell state to one that's allows it to grow and to prosper under nutrient-poor conditions. And so, in order to help validate this project, one of the experiments we did was to look at GCN2 knockout in combination with L-asparaginase. And L-asparaginase basically reduces the presence of asparagine, which is an essential nutrient. And we think that's kind of a model of what a tumor cell may face in terms of amino acid and other sensory deprivation that it has to undergo. And so what you can see here is in the presence of a knockout of GCN2 knockout, this OVCAR8 cell shows very high sensitivity to L-asparaginase, whereas when it's wild type, you can see there's a much less of a sensitivity. And the same result is true also with another ovarian cancer cell, SKOV3. And so what we did to basically using this and other evidence is that we decided to pursue screening of GCN2 using our kinase library. And we had two hits that came about. And we essentially screened at a vendor called Eurofins, which is what we've used for a lot of our kinase projects when they had the kinase available. And we ended up with two weak hits, one OICR 1158 and one 1843. And essentially by overlaying these in a model of GCN2 that we developed in-house, a homology model, we could see that there was actually substantial overlap, yet there were key regions of difference in these two areas, and so that we could actually build upon this scaffold and do some scaffold melding to get to our first novel hit, which was 3337, which was quite a potent GCN2 inhibitor. And so then we continue to develop this novel hit into our lead compound, which is down at 14 nanomolar. And so this compound had very poor PK, and we had to do a lot of medicinal chemistry to convert it into our lead compound 860, which is not only more potent, but it also has really excellent mouse PK. And Methvin Isaac and one of my colleagues, Radek Laufer, were key in being able to develop and do this really excellent medicinal chemistry work to get to this lead compound, which started some very interesting activity. And again, this is asparaginase, and what it does, the chemistry it does, is it actually converts asparagine into aspartic acid, and that leads to the deprivation of this key amino acid, which is essential for cancer cell growth. And so what we found is that we could essentially pharmacologically phenocopy the knockout results that I showed before with 860, the molecule, our lead compound. And we were showing that essentially the molecule doesn't do, the drug doesn't do very much by itself in terms of inhibition, but when it's combined with L-asparaginase, you see a dramatic reduction in tumor cell growth. And so we thought this was a nice set of experiments that kind of further validate the target pharmacologically. And so using the molecule, which has a structure which is embedded in this patent, we were able to actually take SKOV3 xenograft animals and treat them with this drug. And we showed a really nice tumor stasis in this model. And this is as a single agent. And we were able to show that it had nice in vivo efficacy. And so what we're actually doing right now is exploring different combinations, including cisplatin, to be able to demonstrate that there's a synergy with other agents which could be used for ovarian cancer. And so that's an ongoing story, but we're looking to move this project forward. And so now what I wanted to do is shift gears into tell you a little bit about HPK1, which is an immuno-oncology target. And this is a rather complicated diagram, but it's essentially highlighting some of the major players in T cells, which are important to stimulate the T cell function in an immune environment. And essentially HPK1 dampens the T cell response by phosphorylating its major substrate, which is SLP76. SLP76 is really the effector of T cell activity in this context. And when HPK1 phosphorylates SLP76, it leads to its degradation and blunting of this whole pathway. So essentially, if you're inhibiting HPK1, you're re-stimulating the T cell by causing SLP76 to become competent and active again. And so by doing this, we're actually stimulating the T cell independent of the cancer environment, which we think is an advantage or potentially complementary to other immune checkpoint inhibitors, which are basically working very much in context with the cancer cell and the immunosuppressive environment. And so the logic would say that if you have an inhibitor of HPK1, you should decrease SLP76 phosphorylation, you should upregulate this pathway, and you should cause cytokine release, which in this case, the major cytokine that's released is IL-2. And in addition, what we've seen is an increase in T cell proliferation and activity, which I think is a very important advantage of this, not only just the effector activity of a T cell, but also the number of T cells involved too. So we think there's a lot of potential applications of this project, ranging from CAR T cells to stimulation of other T cells and helping them grow ex vivo. So, with all of that in mind, we also saw some additional evidence coming from the literature that said that HPK1 knockout mice were reasonably healthy, and did show all the sort of phenotypes that one would expect from the biological rationale that I just showed. And if you knock out HPK1 or you cause kinase a dead knock-in, you can actually see significant anti-tumor growth. And so, with all of that in mind, we cranked up, and this is an internal project. We weren't working mainly with an academic and collaborator, but we developed what we call a compound progression cascade or testing cascade. And really, without going through all of these boxes in detail, because it's a little overwhelming, I'm sure, it's really showing, I think, the capabilities of everybody on the team and coming together and developing a set of assays to be able to prosecute compounds. So when compounds are first made by the project team, they go into these biochemical assays. And as I alluded to before, we don't do everything in-house. When we can do it externally, we do, because it saves money in the long term to be able to test compounds through CROs. But we had our cell biologists developed assays to measure IL-2 release of Jurkat cells, which are immortalized T cells, and also to be able to extract and to use human T cells, primary T cells, to do analogous set of assays where we're actually looking at cytokine release, proliferation, and the things that we would expect an HPK1 inhibitor to do. And if all of that looks good and the compound passes successfully through this cascade, it goes on into some much more costly, we have to use these assays very judiciously, but we go into in vivo efficacy studies in syngeneic models, which are basically immune competent models where the mouse has the immune system. I'll say a little bit more about that shortly. And I'll also get into another aspect of this program in a minute, which is PROTACs. And we also have a set of assays which is designed to interrogate how well the compound is working and functioning as a PROTAC. So we've made over 2,000 compounds for this project so far. And we've also worked with a good collaborator of ours, Gil Privé, who's at UHN, and he has performed our X-ray crystallography studies for a number of our projects, but most recently for the HPK1 project. And what Gil and his colleagues found was similar to what was already published in the literature, which is that HPK1 exists in kind of an interesting domain swapped homodimeric state, where you're basically one domain is interacting with another domain. And so we actually started our work with a CRO called Proteris, which is an expert in the kinase field. And Proteris actually had to do a little trick to create a construct which was suitable for crystallography, which was to cause a cysteine mutate some compounds, some residues into cysteines, which led to this sort of preservation of this domain swapped homodimer. Ultimately, Gil was able to crystallize the compound without having to resort to these mutations. But what these crystal structures did was enable the medicinal chemistry to help us discern how our compounds were binding, which were binding in the ATP active site as expected, and also to help us to tease apart some selectivity with another kinase, which is called GLK. And GLK is actually 100% homologous to HPK1 in the active site. And that was seen as a real problem with this project because GLK actually functions in the opposite way as HPK1. And so inhibiting fully GLK might have at least if it doesn't have completely the opposite effect of HPK1, certainly it blunts the activity of HPK1, and we see that with some of our bell-shaped curves. So teasing apart and getting selectivity for GLK was a real challenge in this project, but ultimately we managed to succeed by starting again with our kinase library to get back to the theme of the talk, and we screened this library of at the time it was around 600 compounds, and we did follow-up compounds, follow-up IC50s, and we got this. Our initial hit was crizotinib, which is actually an ALK inhibitor. It's a pretty promiscuous compound, but it also inhibits, among other kinases, HPK1, but inhibits LCK at 22 nanomolar. And why that's important is LCK is actually another kinase which does the opposite of HPK1. If you inhibit LCK, it does you no good to inhibit HPK1 because their LCK is actually causing is actually a T cell effector. So you're blunting the effect. So you need to have selectivity over LCK. So our first medicinal chemistry effort was designed to try to show some cell-based activity, but also to get a modicum of selectivity on LCK. And that's what we were able to do with our first compound, 1479. And then we did a lot more medicinal chemistry to show you the evolution of the project. And we got to a compound 20371 through some medicinal chemistry tricks, which I can't go into right now, unfortunately, but allowed us to develop very high LCK selectivity, great PK. And unfortunately, the only problem with this compound is it had poor GLK selectivity. So we did much more medicinal chemistry optimization. We looked at some of the regions around the active site, which differed between HPK1 and GLK, and we were able to design compounds that had good, at least 30-fold selectivity over GLK. We haven't completely eliminated that kinase, but we've gotten up to about 100-fold selectivity in some of our best molecules. But 30-fold selectivity, I think, is giving us a real advantage in terms of being able to show some of the data that I'm about to present. And so, we filed some patents in this area, and a lot of the molecules I'm talking about are coming from Series 3, particularly 35700. And essentially, what we were able to show with our lead compound is that it had a very nice effect in terms of releasing IL-2. And so this is a dose-dependent response of primary T cells to our HPK1 inhibitor, and you can see a nice increase in IL-2, and you can also see an increase in interferon gamma. And different effectors of T cells have, for whatever reason, different levels of cytokine release. And it just turns out that with HPK1, you really see a strong effect with IL-2 compared to interferon gamma. But in addition, this molecule shows the mechanistic inhibition of phospho SLP76, which we would expect at concentrations which are relevant to these other effects, so that we have confidence that we're on target. And it shows some increase in T cell proliferation, which is something gratifying because we've compared our compounds to some of the competitor compounds, and we don't see that increase in proliferation. So we were pleased about that. Another thing is that we saw a little bit of a plateau here. It does bell out at the end, but relative to our early compounds and some of the competitor compounds, which essentially show a sharp spike, they may be very potent, but they show a sharp spike. We're seeing over a few logs a nice increase in proliferation and cytokine release, which we think is a function of having high kinase selectivity. And so going into the in vivo setting, we were able to show that we had nice activity in an EMT6 syngeneic model, which is a breast cancer model. It's kind of been our workhorse model for this target. And we see that as a single agent, PD-1 causes a significant effects, and it works pretty well. And in fact, there's a few complete responses with PD-1 treatment alone at 10 mg per kg twice a week. But then in combination with HPK1, we should see a really nice combination effect where most of the animals had some degree of response, and about half of them showed complete response. And that is that there was no evidence after cessation of treatment that the tumors regrew. So we were really encouraged by this result, along with the fact that we didn't see much weight loss, if any, in this treatment. So we thought that we feel like this is a well-tolerated drug. And so this is another question that may arise as to what is the effect of HPK1 inhibitor as a single agent. And what we see is that it works about the same as PD-1 itself, sometimes a little bit better, sometimes a little bit worse, which in this case, but roughly the same. And so in combination, of course, you see that nice effect of tumor regression. As a single agent, you see significant blunting of tumor growth. So one of the key questions that arises with a project like this is how is it going to be presented in the clinic? And I think there is a role for an HPK1 inhibitor as a single agent if it worked at least as well, and possibly in different tumor types as anti-PD-1, which is the standard most commonly used immuno-oncology agent. And the experiment on the right is basically a demonstration of the fact that when you knock out the effector T cells with an antibody, you essentially see a loss of activity of an HPK1 inhibitor. And all that's really showing is that we're on target in vivo in an in vivo setting. Of course, what we didn't want to see is strong anti-tumor activity in this context, because then one would wonder what is the actual mechanism of the compound. So it does appear to be working through HPK1. As shown in here. And of course, we have all the biochemical data to back that up. So another aspect of this project, which was, we started out thinking about, but then we got into in a more serious way recently, was developing PROTACs. And many of you are familiar with PROTACs, but just briefly to highlight, the concept is you're linking a protein which you're linking an agent which causes a linkage of a protein of interest to an E3 ligase, which ubiquitinates the protein and leads to a degradation from 26S proteasome to cause degradation of the protein. And the general advantages of PROTACs are prolonged PK, because you're degrading the protein, and possibly increased functional selectivity. If you can cause the protein to be degraded selectively, even if the small molecule itself is not a particularly selective inhibitor, then you've got a real advantage. And I think that's something that is very, at this stage, how shall I say it? It's very empirical as to how selective you really are with a PROTAC. But as you'll see in a minute, I think we've got kind of lucky with our series of PROTACs in terms of being able to be selective for HPK1. And now in terms of HPK1 itself, there's some very specific rationale for PROTAC. And it's been shown in the literature that through a couple of different publications that HPK1 has both scaffolding and enzymatic functions. And so possibly you get a more complete phenotype, relative to just standard enzyme inhibitor with a PROTAC. And I think that's something that's often relevant to a lot of different kinases. Maybe the enzymatic function isn't the complete story. So, and of course, we wanted to be opportunistic. We had a set of very nice ligands that were targeting HPK1. So we wanted to be able to see how they worked in the context of a PROTAC. And so we've developed some PROTACs, and one of them is called 39349. And as we had hoped, it actually causes degradation of the protein. This is the y-axis here in this graph is time, and the HPK1 is the levels of HPK1. And this is a Western blot assay in primary T cells. So you're looking at the levels of protein as measured by an antibody. And you see that DMSO doesn't cause any effect. Interestingly, HPK1 by itself does lead to some degradation of HPK1, the stimulation with and without antibody, but not nearly as complete as the PROTAC itself. And so it's a nice assay, and we're very pleased by this result, but it's a very low throughput assay, very time consuming and laborious. So what Richard and his group did is developed a nanoluck assay, which basically is a construct which is HPK1 with a fluorophore to be able to rapidly measure the degradation of the protein. And this can be run in more of immediate throughput context. And so not only can we use this for HPK1, but we can also measure the selectivity of the most important or most challenging off target, which is GLK. And so what we see here is that we do get nice degradation with 39349 and a few of our other PROTACs. Interestingly, compared to the Western blot, you don't see the complete degradation of HPK1. We think that, there's a number of explanations for that. And of course, if anybody else has ideas, we'd love to hear them. But, it's possibly the fact that in this system, the protein is regenerated so quickly, you don't have time to degrade all of the protein. Or perhaps the the residual nanoluck causes some incomplete loss of signal. In any event, what we were very encouraged to see is that GLK, where we basically saw less than tenfold biochemical selectivity in the case of 39349. We saw very nice selectivity and hardly any degradation of this off target. So sometimes you can be dealt a pleasant surprise with a PROTAC in terms of what other proteins it eliminates or does not eliminate in this case. And another encouraging set of data that we found is that 39349 causes a nice time prolonged degradation of HPK1 over not only just days, but actually hours, but multiple days, where it takes at least a week before you get full recovery. And using the facility at Spark at SickKids, we were able to actually do, in a reasonable cost, a few $1,000s, we were able to assess the ability of a PROTAC to affect the entire proteome. And what we saw is the level of degradation of HPK1 was robust and also very selective. If you're not familiar, this is a volcano plot basically showing the significance on the y-axis and the level of the effect on the x-axis. And we're seeing a pretty nice selective degradation of HPK1. There is another protein that's up there called GSK, but it turns out that the fact that it looks like it's close wasn't really truly representative of the fact that it wasn't really being degraded as well. So we've done some additional work on finding new PROTACs, and all this chart is basically showing you is some of the properties of our leading PROTACs, showing that when you incubate over a prolonged period of time, as you might expect, because you're degrading the protein, you get enhancement of the effect, the biological effect you would expect to see. For example, IL-2 release, you see going from 89 to 25, 89 nanomolar to 25 nanomolar over 16 hours. Sometimes the effect is more dramatic in some of these cases, not in this case for whatever reason, but encouragingly, as I mentioned earlier, we're getting very good selectivity on GLK in terms of its degradation. And we're very pleased to see that we're starting to get some mouse PK, which suggests that we might be able to use these compounds in an in vivo setting. We haven't gotten there yet. So we're working on this. We're continuing to work in this area. And so now I just wanted to turn to the last project last little vignette that I want to tell you about in the last couple of minutes, very briefly. And that's HPK, it's the Nuak kinases. And some of you are very familiar with this pathway. This is the hippo pathway. And basically in cancer cells, you see that the hippo the hippo pathway is on and transcription is off, that is a good thing. You want to have that happen, and that's because in a normal setting, YAP-TAZ, which is a key effector of this pathway, is phosphorylated and it's degraded. But cancer cells manage to cause upregulation of this pathway. But what our collaborator Liliana Adesanya at University of Toronto found was that there are kinases which negatively regulate this pathway and cause a upregulation again of transcription through YAP-TAZ. And so essentially, if you inhibit the negative regulator, you again see what you want to see, which is a basal state of hippo being on and transcription, which leading to this upregulation of cancer promoters being off. And so we thought with some nice genetic evidence that this is occurring, as well as some Chewel compound, we set up a project team and formed a nice collaboration between Lil and her group at the Donnelly Center, University of Toronto. Our drug discovery group and Methvin Isaac was leading the charge for the medicinal chemistry. Frank Sakari, who's doing some of the structural biology for this project, and Jeff Rana, who is doing more in vivo models. And so we were able to, use the various skills of this team to work on NUAC inhibitors. And so, of course, at the outset of a project, what you want to do is set up a kind of a product profile so that you know what you're trying to achieve. And it was key that in addition to inhibiting NUAC1 and NUAC2, which we thought was both important for this pathway, we did not want to inhibit some of the kinases that were in the pathway, which would cause the opposite effect. In a number of our compounds, we actually discovered that we're working very well in NUAC, or as inhibitors of NUAC1 and 2 did not work very well at all in cells. And it was only later that we realized they were actually inhibiting some of the kinases in the pathway itself, which would cause the opposite effect, which seems pretty obvious in hindsight. But nonetheless, we discovered the hard way, and we've learned that we needed to screen for and dial out those other kinases. So we've managed to use this sort of guideline to develop compounds, and starting with our kinase inhibitor library hit, another kinase inhibitor library screening effort. We see this compound had WZ4003, has modest activity on NUAC1 and NUAC2, and some activity on phospho-YAP, which is our primary cell-based assay. But it wasn't a novel compound. It had poor selectivity and it had very weak cellular activity. But continuing to optimize this drug into a more advanced compound, 16422, we were able to show that it actually had much more potent NUAC1, NUAC2 activity and a very good level of cell-based activity. Unfortunately, this compound was not at all bioavailable, so a lot of medicinal chemistry effort went into developing a more advanced tool compound, which was orally bioavailable. Unfortunately, this compound, like 16422, caused in vivo activity, and it showed pharmacodynamic activity with respect to YAP staining, which was consistent with this mechanism of action. So we're still working to improve the profile of this compound. Going back to the original set of criteria, we were able to actually dial out Aurora A and PLK, which were mitotic kinases. But there are other mitotic kinases we're still working on dialing out while preserving all these other activities. And so that's a work in progress. But with kinase inhibitors on the active site, of course, one of the key challenges is developing the selectivity you want and actually understanding what selectivity you actually need. And so we've learned several lessons along the way in terms of which kinases are important to dial out, which kinases may not be so bad to have along for the ride. So, that's where things stand there. And so just to summarize my talk, we continue to stay very involved in kinases, and we're very opportunistic, and we'd love to work with collaborators on new and novel kinases that have therapeutic relevance in oncology. And starting with our kinase library, in this case, we've shown how it's been a pretty valuable tool for a lot of our different projects and kinases, because these are compounds that are already designed as kinase inhibitors, so they logically represent a good starting point for dialing in some of the properties you want. And that's where things stand. And right now, I just wanted to finish up by acknowledging all the great collaborators we've had along the way. We had for the GCN2, Rob Rodapello was leading and brought this target to us, and he has a host of collaborators working with him, and including people both at UHN as well as LTRI, Lumenfeld, as well as University of Pennsylvania, NUAC, Liliana Adesano, and Mohammed. And then at Lumenfeld, we have Jeff Rana, Frank Sakari, and some of the collaborators. And for HPK1, our collaborators are Gil Privé, Doug Kuntz, and Neil, who did the X-ray crystallography work that I spoke of. And of course, this is our group that is involved. And the way we work on these projects, we're so interconnected that it's hard to single out different people for different projects. I've done it a couple of times along the way, but really we're such an intertwined team that everybody chips in with a different project. projects. And I think that's what makes it really great to work with these people. So with that, I'll finish up, and I really appreciate all your attention, especially if I've pushed the hour, and be happy to take any questions.
Time for a few questions. Tom?
Yes.
A quick question. So on your kinase inhibitors, have you did a profile to see if they inhibit other kinases?
Yeah, great question. So for our lead kinases, we go to Eurofins, which offers a 58 kinase panel, it's promiscuity panel. So basically they take kinases from different families that are easy to inhibit. And if you can screen against those first, you'll get a good idea as to how overall selective your kinases inhibitor is. And so that's been something that we've used as a tool up front. It costs about $300 to run that US bucks to run that screen. But then if we get really serious about a lead candidate, we'll screen against their entire panel, which is 422 kinases. And so one of the things that's nice is we've worked out kind of a good deal with them. So they give us a bit of a discount, not just an academic discount, but a little bit beyond that because we've been good customers. But so 58 kinases first for drugs that are of high interest, 422 kinases for really important kinases, inhibitors.
For the 422.
It runs us about $60 per IC50. So I think doing the math, it's a few $1,000s. So we can't do that for too many compounds. We have to really be very serious about a compound. Typically, we do that, Peter, when it's going into an in vivo setting. So we want to.
It's activity based or just binding based?
Oh, great question. Yeah, these are radiometric assays, so they're activity based. We also go to DiscoverX, which is actually bought out by Eurofins, and they do binding assays. But ironically, you would think a binding assay would be cheaper to run than an activity-based assay, but it's not. It's actually more expensive. But they do have a few more kinases that aren't covered by the activity-based assays. Yes.
How do you go after the specificity of the PROTACs that you're really excited about? Is it kind of proteomics or?
Yeah, we the proteomics assays that I showed run to the tune of a few $1,000s. So again, we can't be, so we have to use sort of the same considerations we do with small molecules in terms of screening key off-target kinases. But I think we've had the luxury of really going into the PROTACs area in that project after we already developed selective inhibitors. But your question becomes even more relevant if you're starting off de novo with a PROTAC approach. And so I guess my advice or thought for our own team, if not for other people, would be, try to do a fair amount of work to optimize your small molecule before you go into the PROTAC approach, and don't try to rely on the PROTAC approach to rescue you too early on. Yes.
Can you please go a little bit into what guides the decision of whether you are going to pursue the small molecule versus the PROTAC, or if there's any other pharmaceutical modalities that Oyster is working on, if there are any? For example, I don't know, antibody conjugates, unsaturated fatty acids, anything of the sort? Or is it just mostly small molecules?
Yeah, super important point. I mean, I didn't really belabor it too much at the beginning. We are generally a small molecule group. So that's our focus. And, we've traditionally been looking at, drug-drug interactions or enzyme inhibitors as small molecules before we go into the PROTAC approach. And, I can't say that's really necessarily been the best approach. I know that sometimes, we worked on a target called BCL6, which was a protein-protein interaction target. We got some very selective inhibitors of BCL6, and they showed a very mild phenotype. And actually, that was kind of the problem with that approach. And we've just recently learned that Arvinas has discovered PROTACs of BCL6, which have a completely different phenotype, much more robust. We're actually still scratching our heads a little bit about that because we've done knockout studies which have shown BCL6 doesn't cause a huge effect, and yet their PROTAC, for whatever reason, is showing a more robust effect. So that at least opened my eyes that we shouldn't be too we should be more open to PROTACs at an earlier stage than perhaps we have been. And in terms of other modalities, I mean, I think we are looking to expand our sort of our wings in that area, and we'd like to do. I mentioned about radiopharmaceuticals, but we've had a small antibody drug conjugate effort. We haven't done the protein work to be able to develop the antibody drug conjugates. We've been collaborating with our friends at the NRC, who actually have been doing the conjugation work for us and making the actual ADC after we prepare the small molecule in the linker. And we had a nice little project going there for a while. We aren't really superly active on that area right now, but I think that's something that we need to consider to investigate more. And we have a new project with somebody at McMaster, which is actually a covalent modifier of an effector of a macrophage. We're targeting a particular cancer, any particular cancer cell antigen in an effort to bring the macrophage together with the small cell. So it's sort of a small molecule-ish project, but it's actually a little bit more than a small molecule. It's probably somewhere in between a small and a large molecule. So, I think it's a great question. I think we need to be pretty open to a lot of different modalities, but we're not an antibody group. So we don't go and develop antibodies ourselves. We'd have to collaborate with somebody else to do that. But if we have good biology and good targets, why not? And I think that's something we have to be open to, not necessarily with our own skill set, but leveraging our collaborators.
Thank you.
Sure. Yes.
In terms of the medicinal chemistry, is that something that you guys perform, or is that something that other companies collaborate with?
Yes, that's actually what our bread and butter is. We started off as a medicinal chemistry group, and we had no disrespect to anybody, but I think we had a kind of a naive view that being a medicinal chemistry group and embedded in with all these brilliant academic scientists, they would provide all the biology for us, and we'd be able to develop drugs that way. But academic collaborators, they have the ideas, and they have the obviously the smarts to be able to develop drugs, but they don't necessarily have the interest in doing the sort of the more, if you will, menial biological aspects of developing assays, making sure that they're reproducible, running them. I mean, if you're a graduate student trying to get a PhD, why would you want to run an assay on the same target for 2 years? That would be not necessarily in your best interest. So the take home is that we've incorporated did I? Maybe somebody's doing that, and sorry about that. Okay, let me back off a little bit on that. If that's all you're doing, maybe it gets a little bit boring. But in any event, we've developed our own biology group, and we have some wonderful biologists who are very good at rigorous, reproducible biology, setting up and developing the assays. And so I think that's one of the great things about collaborating with our group, is that if you have an interesting target and you need an assay that is robust, our group can help set that up and actually not only set it up, but run it in a routine way. Of course, if we have CTIP funding or funding from another group, then that powers the day-to-day, but that's what we're looking for. So we're all of the different, in that early diagram I showed with all the different disciplines, it all started with medicinal chemistry and went out from there.
I've noticed in the HPK1 inhibitor part, you're quite focused on this GLK potential off target. And it seems like the focus on selectivity is really important to your group. And I'm just wondering, compared to say competitors, it is part of the roadblock for a lot of these small molecule kinase inhibitors that seem really promising in the mouse, but then fail in early phase human clinical trials, because there's not enough focus in the industry on getting the selectivity right?
Well,
or not necessarily.
I think that, industry, it's interesting with a project like HPK1, because we went into it thinking we were the leaders in the field. And of course, at the time, we were among the leaders in the field. One of the disadvantages of a group like ours is if you have Merck and GSK and and Pfizer and all these other groups and biotech groups all jumping in, you find yourselves in a room which is very crowded. And so then, but it does afford you the opportunity to look around and see what everybody else is doing. And it's surprising how parallel our path was to a lot of the other groups. And I think in industry, I think because they can screen selectivity pretty easily, why not? And I think that's something that's done pretty early. So I wouldn't say that that's necessarily the big reason for failure is selectivity within the kinome. Oftentimes, and I didn't get into it in too much detail, in the drug development cascade, you're finding yourself hitting off targets that are completely unrelated to your family of interests, like HERG or something else. And so that's been something that we've had to do battle with, like other groups involved. But selectivity within the kinase family, I think. Generally, groups are getting pretty good at that, both academic, well, mostly pharmaceutical groups, because they can screen so easily.
I have a question about the selectivity too. Nowadays, with the ability to do these in silico docking type experiments, are people employing that approach to address this question of selectivity? Because we probably know crystal structures for at least one or two members of each of those classes of kinases, and clearly there's enough difference in the active site, most of these being ATP site directed inhibitors. There's enough differences in the family that you can see these ranges of selectivity, but are people taking advantage of in silico approaches to try and address that?
Yeah, one of the great advantages of having a computational chemistry group right down.
Notice Genna's not here in your picture.
Genna's not here in the picture, and that's because he's not. He didn't make it. It was a Friday actually, and it's unfortunate. One of my things I'm going to do for when our next picture, I'm not going to have it on a Friday because a lot of the people were gone on that particular Friday. I'll have it on a Tuesday or Wednesday. But anyway, Genna and some of the other great computational chemists in our group, having them around, the first thing we do is, okay, we have a problem with this off target. Can you help us understand it? And then let them start to do their magic in terms of developing a homology model and being able to actually predict whether we can actually get selectivity and how we can get selectivity by interacting with or clashing with key residues. So we do use them all the time. It's a part and parcel of what we're doing.
So, but do you see at least at some point that becoming a more capable approach to identifying molecules that are going to have selectivity than to actually roll up your sleeves and do the screens?
Yeah, I think we have to be very mindful of the fact that it costs a lot of money to do these screens.
Yeah.
And if we have a computational chemistry that can help us up front work things out, then that's really an advantage. And so, having a crystal structure of the off-target kinase can be really valuable. And so with some of the projects, we've been able to, for example, get crystal structures of PLK and Aurora so that we could see what clashes and how to design inhibitors that were selective, at least against those targets. And being able to design drugs that are selective for one, oftentimes, unfortunately, at least I feel like we're still at the point where, oops, we're inhibiting some kinase we didn't know about. Now we have to focus on that. So then we have to apply our computational chemistry and all our empirical resources to developing selectivity for it. Wouldn't it be nice to be able to develop a de novo, figure out ways to design compounds that, and I think that there's going to be advances in that area, but I don't think we're quite there yet.
Well, what I'm hearing though is, although crystal structures are not available for all of the human kinome, probably what, 10% of them or something like that. But people are going to AlphaFold, and so do you believe that the structures that are generated with AlphaFold are going to be suitable for doing this kind of analysis? Yeah, I think I'm really been very impressed with the latest version of Rosetta. The time the computational those AI techniques to generate structures are really coming a long way, and I think in a few years those are going to be
really close to being as good as the crystal structures. I mean, it's just maybe I'm being overly optimistic,
Well,
but
I'm just thinking, can you think of an example of a good inhibitor that was developed starting off with that technology? Not yet.
not yet. I don't think so. Stay tuned.
If there are no other questions, then please join me in thanking David for. Hopefully we won't get stuffed on tonight.